So now that we understand how the process of gene expression works, we now are positioned to start talking about how gene expression is regulated. And I'm going to divide this up across a series of 3 videos. In the first video, we'll examine short-term methods for regulating gene expression in prokaryotes, because these are relatively simple systems that will give us some ideas that we'll then take to look at the regulation of gene expression in eukaryotes. And from there, we will be positioned to start thinking about the regulation of gene expression over longer time frames. So for example, we'll focus on expression during embryonic development in the third video. But before we get into this topic, I want to take a moment to just talk about, what's the big deal? What can we better understand from having a good grasp of how gene expression is regulated? So here's a couple of different ideas for why it's useful to understand how gene expression gets regulated. So first off, if you've known someone who has cancer, cancer generally involves changes to gene expression in the cancerous cells. So if we understand how gene expression is regulated, we can make sense of the changes that happen to gene expression in cancerous cells and potentially figure out how to thwart those cells. Second, we know that people that suffer from drug addictions experience a lot of long-term changes in gene expression. And that's part of what makes drug addiction a really persistent, difficult problem, is that it's linked to these long-term changes. So again, if we have a better sense of what's happening in terms of changes to gene expression, we may be better equipped to help people that are suffering from drug addiction. And then a third reason why is changes to gene expression are tied to learning and memory as well. So the process of learning and remembering things also involves shifts in gene expression. And interestingly enough, some of those gene expression changes are actually what's called epigenetic. So they're changes to gene expression that get inherited across a couple of generations. And that really threw biologists for a loop when they first started thinking about that, because it's very different from thinking about everything being encoded in DNA. So it turns out that no, there's other sources of information in cells that get inherited across a couple of generations. And a major example where we've seen a lot of evidence for this type of change in gene expression comes from studies of maternal stress, where if a mother, particularly well studied in mice, if a mother mouse experiences an inordinate amount of stress over her lifetime, then her offspring will actually show different patterns of gene expression compared to a mother mouse who did not experience that level of stress. So having a better understanding of how gene expression works gives us tools for thinking about how to address some of these major issues in biology and in health. We're going to start out talking about gene expression in prokaryotes, in the bacteria, because in this case they have a simpler method for regulating gene expression, and that's going to give us the mental tools to then think about how these processes have gotten modified in eukaryotic organisms. So we're going to work with E. coli bacteria, a type of E. coli that are found in our guts, and we'll start out thinking about one particular substance that gets used for a lot of things in bacteria, tryptophan. And tryptophan is actually a non-essential amino acid for E. coli. So what that means is that E. coli can actually make their own tryptophan. So if there's no tryptophan present in their environment, no problem, they can make it. So it's not essential. In contrast, we can't manufacture our own tryptophan. So if we eat foods that don't contain any tryptophan, we will actually wind up getting our tryptophan from the E. coli that live in our guts. So pretty cool. But so this is one of the classic examples for thinking about gene expression regulation. So the production of tryptophan in E. coli is actually regulated in 2 different ways. One of those mechanisms is through feedback inhibition. So we talked about that quite a while ago. Now we're seeing an example of this. So basically, if tryptophan levels build up in a cell, the buildup of tryptophan will actually come back and it will inhibit one of the enzymes that's involved in the pathway for synthesizing tryptophan. So that's one mechanism. The other is that tryptophan itself can also regulate expression of the genes that code for these proteins or enzymes that manufacture tryptophan. So you can imagine, so tryptophan, if it can block gene expression of these genes when it builds up, what that means is if you have a bunch of tryptophan around, the production of these enzymes will drop and you won't wind up shifting everything into making more tryptophan. And I do have a note on this slide. You do need to be very familiar with the regulation of tryptophan gene expression is one example, and then we'll also examine lac gene expression later on as well. So how does this regulatory process work? Well, it works by thinking about the gene region and some associated regions as well together as being involved in the regulatory process. So this whole thing here is referred to as an operon. That's the gene and the regulatory region associated with it. And in addition to the section of DNA that codes for either for the genes themselves or for some of these regulatory sequences, there's also another gene found somewhere else in the E. coli genome that's called a repressor gene. So this repressor gene is always expressed at a low level and produces a protein referred to as a repressor protein. And we'll get into that more in a minute. In this regulatory region, there are two things that we're going to focus on here. One is the promoter, so where RNA polymerase binds. That's found within this regulatory region. And then there's a new region associated with that now called the operator region. And we'll talk more about that in a moment. So first, let's set the stage by thinking about kind of a default condition for the trip operon. And let's consider this to be a situation where there isn't any tryptophan present. And so in that case, the cell's going to want to make its own tryptophan. And so this operon is going to be on and functional. And what I mean by that is that RNA polymerase is going to bind to this promoter, and it's going to be able to transcribe the genes that code for those enzymes that allow the E. coli to manufacture tryptophan. Okay, but now let's say that there's a change in status. So maybe you eat a large meal. Turkey is a classic one that contains a lot of tryptophan. And now there's a bunch of tryptophan in your gut. And so the E. coli in your gut now have access to abundant tryptophan in the environment. So at this point, it doesn't really make sense for an E. coli cell to keep making more of its own because it takes energy and resources to make those enzymes and then to make the trip itself. So it'd be really beneficial for bacteria to be able to shut off this process. And they can do that by using a molecule type called a repressor. So that's that repressor that I was showing you earlier. Along with that, the repressor can't bind directly to the operator by itself. If it could, we'd run into problems because it would be impossible to express this gene at any time. So instead, the repressor also requires the presence of a co-repressor that will act as an allosteric activator. So it'll turn on this repressor and allow that repressor to bind to the operator. And I have a question here about whether you can think of a good candidate for a co-repressor. I'll give that away. The good candidate is tryptophan. So when tryptophan levels build up in the cell, tryptophan can bind to this repressor protein. The repressor protein can then bind to the operator, and that's going to then block the binding and transcription of these genes by RNA polymerase. So collectively, what's happened here is through the action of this repressor binding, this process is then referred to as repression. So you don't get messenger RNA production, we don't get enzyme production, and we get a drop-off in tryptophan synthesis. So here's just a little cartoon that overviews that whole process, because what I want you to be aware of is that this is kind of a dynamic process happening in a cell, right? So you might wind up in one situation where there's no trip present, so at that point the repressor is going to not be able to bind to the operator, so RNA polymerase is going to be able to transcribe. It'll produce messenger RNA that's going to get translated into these proteins, and then that means these proteins can then catalyze the reaction that produces more tryptophan. What that then means is now we're in a status change situation where tryptophan is present, and so where it makes sense for an E. coli cell to now repress this process so that tryptophan will bind to the repressor, the repressor will bind to the operator, and now RNA polymerase can't transcribe, and whatever tryptophan is around will get used up by that E. coli, and eventually it'll wind up back in a situation where it doesn't have any tryptophan. And so this is really a dynamic process where the state in a given cell will change depending on the levels of tryptophan that are present. And that allows an E. coli cell to make tryptophan when it needs it, but to shut off production when it's unnecessary. So we refer to this style of gene regulation as negative gene regulation because it's really an on-off regulatory mechanism. So I like to think of it as being kind of like flipping on a light switch on or off. And negative gene regulation is defined by the presence of a repressor protein that can switch a gene off. And there's actually two different types of negative gene regulation that can happen. So the first one that I showed you, repression, involves the presence of a co-repressor, and together those activate the repressor, and that causes the gene to be shut off. But there's a second type of negative gene regulation that's referred to as induction. And during induction, basically, if a particular substance is present, that substance will bind onto a repressor that can bind to the operon by default and will pop that repressor off and turn the gene on. And so this relies on allosteric inhibition. So let's take a look at an example of induction to contrast with the example of repression that I just showed you. And so here's our example with another operon referred to as the lac operon. So to understand this operon, we have to think a little bit differently about the players that are involved. In this case, this is named after lactose, which is a type of sugar. So I've illustrated it up here. And this type of sugar is a really great energy source for E. coli. So when there's lactose around, it's beneficial for E. coli to produce enzymes that will allow the E. coli to break down that lactose and use it for energy. And it requires a set of specific enzymes to transport that lactose into the cell and then break it down. So you don't need to know the names of these enzymes, but just for posterity's sake, that's beta-galactosidase, permease, and transacetylase. So in some ways, the DNA sequences look fairly similar to what we just observed for the trip operon, but there's a slight distinction here instead. So we've got a gene region that codes for the lac repressor protein, and when that lac repressor protein is present by itself, it can bind to the lac operon. And we've got the same type of thing here where you've got a promoter region and then a gene region, and when this operon is on, it's possible for RNA polymerase to transcribe that gene region that will get translated from the messenger RNA, and you'll get the production of these proteins. Okay, so under conditions where there's no lactose present, that is the condition in which this repressor protein will be bound. And so it will prevent transcription when it's not very useful to be producing these proteins. And in contrast, when there is lactose present in the environment, lactose will act as an inducer molecule by binding onto the lac repressor, and at that point it causes a shape change that causes the lac repressor to pop off of the operator, and that's going to allow RNA polymerase to bind and to transcribe this gene. But I want to remind you, this is still an example of negative gene regulation because it's an example of on-off regulation, right? So when the repressor is bound, the gene's off. When the repressor is not bound, the gene is on. So that might then have you wondering, if this is negative gene regulation, then what constitutes positive gene regulation? And for positive gene regulation, in contrast to this on-off status change, positive gene regulation involves molecules that allow a cell to ramp up its production of any particular set of genes. So in this case, we're actually going to look at the lac operon again because the lac operon can also be positively regulated. And so to make sense of this, I need to talk a little bit more about E. coli metabolism. So lactose isn't the only source of energy that an E. coli bacteria can use. E. coli actually prefer to use glucose because glucose is a lot simpler to break down. However, if there isn't any glucose around, then E. coli will use lactose instead. And the positive gene regulation for the lac operon is a mechanism that allows an E. coli cell to hyperactivate the lac operon when lactose is the only energy source that's available. So here's how that works. It involves two more molecular players. Those molecular players are abbreviated CAMP or cyclic AMP, and then CAP, which is just the cyclic AMP activator protein. So when glucose levels are really low in a cell, one of the consequences is ATP gets used up, and one of the byproducts when ATP is used up is cyclic AMP. So if a cell has really low glucose levels, then the amounts of cyclic AMP that are present will start to build up. The cyclic AMP acts as an allosteric activator of CAP. And so when cyclic AMP binds to CAP, that activated CAP then binds to another region of the lac operon that's actually upstream of where the promoter sequence is for RNA polymerase. And it facilitates binding of RNA polymerase and transcription. And so what that means is if cyclic AMP starts to build up, CAP gets activated, it binds, and that's going to ramp up even more transcription and translation of the genes in the lac operon. However, I'd note, so if glucose and lactose are both available to an E. coli cell, then there won't be very high levels of cyclic AMP, CAP will not bind, and so we'll get some transcription of the lac operon, but not nearly the same amount. Okay, so that brings us through short-term methods for regulating gene expression in prokaryotes. So in the next video, I will talk about the regulation of gene expression in eukaryotes and kind of how we go from these simple on, off, or up systems to more involved and sophisticated ways of regulating gene expression.